right now. In chapter 12, it's all about the brain. And we can't possibly go over every minutia, every detail, but we can do a lot of it. So let's start at the beginning. I will pull up my first whiteboard and I'm going to begin with the term cephalization. That word, cephalo, means head. So whenever you see C-E-P-H-A-L, just think head. Cephalization refers to the process, the developmental process of major brain structures like the frontal lobe and the accumulation of billions of neurons in the central nervous system, AKA the development of the brain, basically, both in utero, embryologically, it's a process. It's also an evolutionary process. When we look back in time, the complexity of animals and specifically mammals, there's been higher cephalization, obviously in mammals like humans, primates and humans. So cephalization just refers to that developmental process of both the frontal lobe and the accumulation of neurons in the head. All of that begins with something called the neural tube. So all central nervous system, that's supposed to be an L, the neural tube, all central nervous system tissue, when we are developing embryologically, develop from a structure called the neural tube. That includes the brain and the spinal cord. So the neural tube is that embryonic structure from which the brain and spinal cord develop. And there's three primary sort of regions. They're called vesicles that appear. Three primary vesicles that appear during development, embryologic development. And what they are basically is just outcroppings or bulges in that neural tube. And those three are the pros, P-R-O-S, and cephalon, the mes, M-E-S, and cephalon, and the rhomb, R-H-O-M-B, the rhomb encephalon. That refers to these bulgings in the neural tube. And these give rise to areas in the brain. They're sort of primitive developmental areas of final brain areas. So for example, let me write this out here. There is a specific figure in your book which goes into more detail about this. It would be 12, it's figure 12.1, the first one on the first page. And it tells what all of these vesicles eventually develop into. The first stage would be the prosencephalon, and I believe the literal translation is just forebrain. Mesencephalon is mid. And finally, the rhombencephalon, that means hind or backwards. So H-I-N-D, the hind brain. And those areas then develop further into sort of adult brain structures. That can be found in figure 12.1. So I do want you to, this is just the beginning of figure 12.1 that I've listed. I want you then to look at the second half of it and know that like the cerebrum, cerebral hemispheres, cortex, that all develops from the prosencephalon. The midbrain eventually develops into most of the brain stem structures, like the midbrain, the pons, the medulla oblongata, as well as the cerebellum. And finally, the rhombencephalon basically also participates in brain stem structures. And what's not included in this business here? So the neural tube, when it's just it's a tube of tissue like this, and then it eventually embryologically develops into these bulges like this, sort of a top bulge, a a mid-bulge, and then these three structures that I've listed here, here, and here eventually go on to become brain structures. What's left of this neural tube eventually becomes the spinal cord. And that's how all of this central nervous system business begins to develop. I'm going to briefly talk about brain ventricles before I go any further. I'm going to go to a new page, but I'm going to write this word. The ventricles of the brain are cavities or spaces. They're major cavities that house cerebrospinal fluid. So they contain a fluid that helps bathe the brain, keep it moist and protected. Those ventricles they develop in very specific ways. And that's what I'm going to quickly draw on the next page. So if this is a human brain that I'm drawing here with my black pencil, there's the cerebellum. Here's the brain stem down to the spinal cord. The ventricles are pictured in your book somewhere. They are literally spaces containing fluid sort of in the center of the brain. And they look something like this. This is a lateral view, it's a side view. And there are two ventricles that are in tandem to each other, one left and one right, but they're toward the center of the brain like this. And these two lateral ventricles are shaped something like what I'm drawing here. Then there's another ventricle, another major cavity or space that has a funny shape to it and a hole in the middle of it. We number these ventricles. These would be ventricles 1 and 2. Ventricle 3 is this one that I've drawn here. And then there is a tube, a fluid-filled tube called the cerebral aqueduct that runs down here. And we eventually get something called ventricle number 4 that kind of looks like a tail on this structure. So there are 4 major ventricles of the brain. This then eventually continues down the center of this spinal cord. Those are the 4 major ventricles. And by the way, we call ventricles number 1 and 2 the lateral ventricles because they're in tandem. They're 2 parallel right next to each other. These contain that fluid, cerebrospinal fluid, and I'll tell you later how that develops. That fluid circulates throughout these ventricles. It goes down the center of the spinal cord. It makes its way out around the outside of the spinal cord and comes up and circulates all around the brain like this. That is cerebrospinal fluid circulation pattern. All right, now I'm going to talk about specific areas of an adult brain, of a fully developed brain. And we're going to start with the cerebral hemispheres. When you hear that word cerebral hemispheres, those are the 2 big sections, large sections of the brain that you and I call the brain. So I'm just going to cut really quickly and hold it in my hands. If I take my 2 fists and I put them together like this, that's roughly the size of a human brain, the cerebral hemisphere in the brain. And the reason we use this as a model, our 2 fists, is because there is a crease, something called a fissure, running down the center of the right and left cerebral hemispheres. So real quickly, I'm just going to write, get my black pen back. Those cerebral hemispheres are the big portions. It takes up about 80% of brain volume. And they are right and left hemispheres. That makes up 80% or more of brain mass. And then there are terms that we just need to go over, anatomical terms. A gyrus, G-Y-R-U-S, is a raised ridge on that surface of the cerebral hemispheres. We've all seen pictures, at the very least, of what a human brain looks like. Those raised bumps or ridges all on the surface are called gyri, that's plural. A gyrus is singular, so technically I should probably take the S off of that for a gyrus. A sulcus, S-U-L-C-U-S, is a groove. So you know the brain surface kind of looks like this. There's raised ridges and then these invaginations or grooves if we look at the surface of it. This would be a gyrus and this would be a sulcus. A fissure is just a deeper groove than a sulcus. All three of these features greatly enhance or increase the brain surface area, especially of the cortex region. The median longitudinal fissure represents the crease between my two fists. If we look at a brain from the front, fully forward, I'm not going to draw all the individual gyri and sulci like this. The human brain from the front has this, it's called the median longitudinal fissure that separates the right and left hemispheres of the brain. I'm going to abbreviate longitudinal. Then there is something called a central sulcus and lateral sulcus. So from the side, this is a lateral view of the brain. There's the cerebellum, brainstem. From the side, we have something called the central sulcus, which comes down this way on both sides. It's behind where your hairline is, most likely. That separates sort of the frontal lobe up here from the two parietal lobes back here. And there's something called a lateral sulcus, which runs on either side, both left and right, kind of like this. So I'll label these. This would be called the central sulcus. And there's the lateral sulcus right in the lateral sulcus basically separates what you and I would call the temporal lobe from that frontal and parietal lobe. It runs this way. And now let's just define cerebral cortex, because we're going to be talking about a bunch of different cortexes. That should have an L, cerebral cortex. A cortex is sort of the outer covering of neural tissue on the brain, on the hemispheres, but it has a thickness to it. So when I say outer covering, it's not thinner than a piece of paper. Picture this: if your whole head was a brain, and you put a bike helmet on, or a motorcycle helmet on, or a football helmet on your head, the cortex is analogous to that helmet. So it's the outer covering. It has some thickness to it, but the cortex is really the tissue that's on the outside of the brain. So let's go to a new page, and I'm going to write cerebral cortex. Now that we know what it is, it's sort of that outer covering of neural tissue. This is where consciousness exists. So I'm going to write that out for you. It's where we experience things like sensations, communication, memory, learning, understanding, awareness, all of the complex things that we think of when we hear the word brain. It's also where things like voluntary skeletal muscle movement originate. Now, within central nervous system tissue, particularly in the brain, also in the spinal cord, I think I defined this earlier for you, but I'm going to remind you. Gray matter versus white matter. It used to be spelled M-A-T-E-R, but lately I've seen textbooks spell it with 2 T's. Remember, gray matter is unmyelinated neuron cell bodies. and white matter is myelinated neural tracts. The cerebral cortex tissue is made up completely of gray matter. It's unmyelinated neuron cell bodies. That's where processing of information goes on. So there are no myelinated tracts in the cerebral cortex. The cortex is comprised of literally billions of neurons. Now we know they are complete gray matter, unmyelinated, mostly neuron cell bodies. It's roughly about 4 millimeters thick. Depending on what area you're talking about, it can thin out a little bit. So that motorcycle helmet analogy I gave you, or football helmet analogy, it's really only about 4 millimeters thick, but that's significant in thickness. It makes up like 40% of brain mass, I think. And those invaginations, those sulci and those gyri, those raised ridges, they increase that surface area about threefold. It like triples the brain surface area. All neurons in the cerebral cortex, I'm just going to write in the cerebral cortex CC, are classified as interneurons. So they're not sensory afferent neurons. They're not motor efferent neurons. They are all interneurons, those association neurons. All right, let's move on. I'm going to now start identifying different cortexes within. This was a generalized term, the cerebral cortex. There are certain areas that we identify into defined cortex regions. So let me draw. I'll use this drawing that I've started sort of. This would be the left side, looking at the left side of the brain. There's the cerebellum that hangs down below the occipital lobe. So that's kind of what the brain looks like from a lateral side view. Here's the brain stem. I'm going to use this diagram to identify specific structures, and right now we're doing cortexes within the cerebral cortex. I'll try to stick with the colors that your book uses to help you. The first cortex we're going to identify is called the primary motor cortex. Everything in red in your textbook is part of the motor cortex, the generalized motor cortex. The primary motor cortex, one with a degree next to it, means primary. And we're only looking at one side of the brain here. That goes down the other side. We're looking at the left side, left hemisphere. That would run over sort of, if we were to look at a brain from the frontal view, the primary motor cortex goes up, there's the top, and it goes down the other side as well. It has a thickness to it like this. That's a frontal view. This specifically controls voluntary skeletal muscles. When you raise your hand, take a step, move your arm, anything that's done to voluntary skeletal muscle is under the control of this primary motor cortex. It contains nerve cells called pyramidal cells, which get their name from the shape of the cell body. They look like pyramids. Those pyramidal cells from this cortex, remember, this is a motor efferent area that's sending commands out to skeletal muscles. But anyway, those pyramidal cells, their tracks extend. I'm going to draw a little pyramid up here. Their axons, their tracks extend down into the spinal cord like this, and they're sending signals this direction away from the central nervous system. Also, motor innervation is contralateral. That means The left side of the primary motor cortex controls muscles on the right side of the body, and vice versa. The right motor cortex, primary motor cortex, would control skeletal muscles on the left side of the body. There's also something called the premotor cortex, which sits right in front of, it's an area right in front of the primary motor cortex like this. And that premotor cortex helps plan for fine motor movements. It just assists the commands that come out of here. So I'm drawing an arrow this way. Also within this motor area, on the lateral side, but located only on the left hemisphere, it's not on both sides, is an area right here. I'm dotting it because your book does it as well, but it's still red tissue, so it's still part of the motor area. It's called Broca's area. I'll label that. Broca's area specifically is in the left hemisphere only, not on both sides, and it controls speech articulation. That means it helps us pronounce and actually speak words. So it's a highly developed motor area. How we know these things is usually from when somebody has suffered damage in a specific area of the brain. Then scientists, neurologists look to see what the result is of that damage. So somebody who has damage in Broca's area, typically they can think of the right word to say, but they have trouble actually articulating it, saying those words. Next, I'm going to do the primary somatosensory cortex, which is in blue. So I'm going to stick to the same colors as your book. This would be the primary, sorry, getting ahead of myself with my spelling. The primary somatosensory cortex is located just posterior. It does the same thing. It loops around both sides on both hemispheres, but it's behind the primary motor cortex, sits behind it. It receives information. It's a receiving center. Receives info, I'm just going to put from sensory receptors. Okay. Mostly from the skin, but also from proprioceptors in our joints and stuff. So when sensory receptors are monitoring our external environment, like receptors in our skin, touch receptors, temperature receptors, yada, 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 those impulses get sent to the primary somatosensory cortex like this. And this area receives info from those receptors. And the somatosensory association cortex, which is located just behind it, it's let me get my blue pen back. It's an area right here. It's a little bit bigger. That's called the somatosensory association cortex, or area. That's behind the primary somatosensory cortex, posterior to it. It helps integrate sensory input, that stuff like temperature and pressure from those receptors, to interpret how something actually feels. So I'm going to put that. So it assists this area here, right in front of it. These somatosensory areas help us discriminate or spatial discrimination. Okay, let's move on. Next is the primary visual cortex. So remember where the occipital bone is. The occipital bone is located back here on the back side of the head. I'm just going to darken it in for you. We call this whole area the occipital lobe. Back in the occipital lobe is a very specific area called the primary visual cortex. I only have one tone of blue. Your book, yes, highlights it in dark blue, so I'm going to color it in. That is the primary visual cortex. So let me go down here. It's in the occipital lobe. That is the specific area of the brain where we interpret what the signals are from our eyes, the stimuli from our eyes, and that's where we sort out actually what we are looking at or what we are seeing. So it interprets information from the optic nerves that emanate from the back of the eyes, which would be way up here. So if our eyes are located right about here, the optic nerves travel all the way back here and they actually crisscross, but they eventually end up, the impulses of those nerves end up way in the back of the head. That's where we determine what we're actually looking at. A person, a tree, a car, that all happens back in the primary visual cortex. The primary auditory cortex is not highlighted very well with a color, so I'm just going to pick a new color. The primary auditory cortex is located in the temporal lobe region right about here. That is where we interpret. It receives impulses. This part of the brain receives impulses from something called the vestibulocochlear nerve, which emanates from the inner ear structures. And it interprets what we hear, the specific sounds we're hearing. For example, that sounds like something hard being wrapped against a hard surface, a knocking sound. I make those interpretations of what I'm hearing right here in my brain at the primary auditory cortex. Also, I'm just going to write hearing. It's interpretation of hearing, equilibrium as well. The olfactory cortex is located also near in that auditory region in the temporal lobe. So I'm going to just write that in black. I'm not even sure if your book, I don't see it on the picture, but the olfactory cortex is also located in this area as well. So I'm just going to draw an area up here. That's just where we interpret what we're smelling. So it's actually sensory receptors, for example, in our nasal cavity that sense changes in the chemical environment of the atmosphere. And then those impulses are sent to the olfactory cortex where we interpret, that's a pleasant smell or an unpleasant smell. The gustatory cortex, which is the sense of taste, is the same area. Let me use a different color. I'll use yellow. That's the sense of taste. That's where we interpret what we're tasting. So that all the auditory, olfactory, and gustatory cortex are all located in the same temporal area. All right, I've made quite a complicated diagram here. If you need me to refer back to it, I'll try to save that, but I'm going to go on to a new page. And just to remind you what the flow of sensory information is, let's do that. Sensory input comes into the brain via, well, it starts at sensory receptors. So we have receptors located either internally or very close to the external surface of the body that send then signals. They sense some sort of change, either internally or externally, and they send signals to primary sensory cortex. Predominantly that's somatosensory cortex. And then those sensations go to that association area behind it. And then there's something called, then that's a signal gets sent to what we call multimodal association cortex. And this is what actually gives those sensations meaning. Puts things into context for us in our brains. And then it is in these association cortexes where a decision is made to do something or not to do something about the original change that was sensed out here. All right, so let's keep moving with brain structures. The next term that I jotted down on my notes is and that I wanted to review with you is this. This is a new topic. Diencephalon is how you pronounce that. This refers to the inner area and structures of the brain. So if we were to, I'm going to go back to one of my previous drawings and see if I can help you make sense of this. This diagram basically went over what we could see in the cortexes, kind of on the surface of the brain, if you will. It's the outer covering. If we go further back, and if I were to make the cortex invisible, the diencephalon, I'll use a different color, just refers to the stuff right here in the very center of the brain that we can't see from the outside at all. We'd have to strip away a whole lot of tissue. We can see some of those structures from the very most inferior surface of the brain, but by and large, it's stuff located deep inside of the brain that we can't see. So we're going to strip away all of this material and look at this, the diencephalon, okay? So let's do that. There are certain structures within this inner area that I'm going to go through like a laundry list and identify them. The first one is I'm trying to go in the order of your book. And I don't know why I jotted down hypothalamus. Let's do the thalamus first. These are two on the inside of the brain. There's two egg-shaped structures. If we were to look at from the frontal view, they'd kind of look like this, and they're attached by this little tiny mass right here. That's for a front view. A side view, they sort of look like egg-shaped structures like this. So they're almost in pairs. The thalamus is an area, a structure, specific structure that receives, it does some interpretation, sorts incoming information, and then sends it to the appropriate cortex. So some books refer to the thalamus as the editing center of the brain. That means information comes into the brain, and this structure, the thalamus, sort of decides what to do with that information. And so information comes up the spinal cord this way. There's actually the brain stem here. Information comes up in this direction. That information heads to the thalamus region, and it's the thalamus that says, okay, we need to send that incoming information to the motor cortex, to the somatosensory cortex, way up to the frontal cortex, so on and so forth. So that's why we call it the editing center of the brain. Just below that structure, I'm going to a new page now. If this is the thalamus from a side view, just below the thalamus is an area or structure, and it kind of comes to this weird W shape like this, called the hypothalamus. Hypo means below. The hypothalamus is even more complicated than the thalamus in its functionality. It contains nuclei or clusters of cell bodies. It's densely packed with cell bodies. This is the major neuroendocrine gland of the human body. It's made of both neural and endocrine tissue. So it has neural tracts and secretory cells that can secrete hormones as well. It's a dual organ. It's wildly complicated. If I have time, I'll go into more of its specifics later, but suffice it to say, this is the area that regulates things like hunger, thirst. It's also the thermometer, so I'm going to write body temp. Sleep cycles. And the list is much longer than that. It also controls or regulates the pituitary gland, which is a major hormone secreting structure that hangs down from the hypothalamus like this. So this would be the pituitary gland, and it secretes 6 major hormones. So this thing up top, the thalamus, is just an editing center, sorts information and sends it somewhere else for interpretation. The hypothalamus is a major regulatory control center of the entire human body. And that hypothalamus also controls, amongst other things, it controls this structure, the pituitary gland, as well. All right, I'm trying to keep my picture as uncluttered, but as clear as possible. Below, let's do this. Hypothalamus, we said, regulates hunger, thirst, body temp, sleep, pituitary. I'm going to explain to you really quickly how it regulates something like thirst. There are thirst centers located in this structure. Let me color it in so we know what we're talking about. I'll make it yellow. Hypothalamus is now yellow. We have thirst centers in the brain right there. It also triggers the release of a hormone called ADH that tells our kidneys to reabsorb more water. So let's just say you haven't had anything to drink all day or even for 2 days. We can't go without fluid for much longer than a couple days, 2 or 3 days. What happens is there are thirst centers in the hypothalamus that are triggered to really make us want to drink fluid. It's a life-surviving mechanism. There's also a trigger of the release of a hormone called ADH, antidiuretic hormone, which goes to the kidneys and tells our kidneys basically not to urinate, not to secrete fluid, because we need to hang on to that fluid for a survival mechanism. So when you haven't had anything to drink for a long time, the reason that your urinary system is not active is because the hypothalamus is telling the kidneys to hold on to every drop of fluid that it possibly can. It's a survival mechanism. The epithalamus, so let me go on to hypo means below. So here's the thalamus, hypothalamus is below it. Epi means above or around. This is a less defined area, sort of, let me use a different color, sort of out here. If I'm facing this direction, the epithalamus are structures located in this region above and beyond the thalamus. So I'll underline that blue. This specifically contains an area called, or a structure called the pineal gland, or pineal gland, some people call it, P-I-N-E-A-L. Remember, we're deep in the center of the brain right now. That pineal gland or pineal gland secretes a hormone called melatonin. probably heard of it. Its main function, it does have something to do possibly with how we feel, mood type of hormone, but its main function is regulating our sleep cycles. So it's dictated, this gland and its secretion is dictated by sunlight, or the lack of sunlight. And then this hormone that's secreted from the pineal gland, which is located right about here, that melatonin that is secreted from this little tiny gland acts on the hypothalamus to regulate our sleep cycles when we're tired and when we feel tired and when we feel wide awake. Now I'm going to go down into structures of the brain stem. So let's do a new picture. So quickly, here's the brain. Here's the cerebellum. Here's brain. That diencephalon region, I'll color that. Let's just color that in blue. Here's the thalamus. Here's the hypothalamus, epithalamus, pituitary gland. Now we're going to go down into the brain stem, this area here. It's considered part of the central nervous system and technically part of the brain itself. The spinal cord actually begins below the brain stem. So this would be spinal cord. There are three main regions to the brain stem. They're pictured in your book and they're shaped something, I'm going to redraw my drawing, they're shaped something like this. There's a bulge egg-like structure here. Above it is this sort of funky hook-like structure. It goes like this. So this would be region 1 of the brainstem. This would be region 2. I'll label them in a minute. And region 3 is this bulge below region 2. We call this most superior region of the brainstem the midbrain. Just anterior and below, inferior to the midbrain is something called the pons, P-O-N-S. That's this egg-shaped structure. It's all connected. That's why I didn't necessarily break it apart from that. And then finally, below the pons is the medulla oblongata. Those are the three structures of that brainstem. The midbrain, I'm just going to give you some laundry list, a Reader's Digest version of the functions of this. The midbrain basically is involved with pain suppression, fear suppression, some eye functions, and hearing pathways. So I'm just going to write eye, ear for hearing, fear and pain suppression. The pons is mostly a neural conduction pathway. So it's mostly white matter, which is myelinated neural tracts. Just think of transmission pathways. It's mostly white matter. For pathways connecting the medulla, its neural pathways is sort of a connection pathway like this, downward and upward as well. The medulla, though, has much more specific functions. I'm just going to write transmission pathways. I am going to ask you about these structures on the next quiz, so it's a good thing you showed up for this. The medulla oblongata in its function contains, first of all, it contains a crossover area where things go from left to right and right to left from the cerebral hemispheres. Remember we said there are contralateral actions. Well, that crossing over, a lot of it occurs not up here in the brain, but right here at the medulla oblongata, we get crossing over of those neural pathways. It also contains, so I'm going to draw an arrow over here just because I need more space. The cardiovascular center is located in the medulla. That regulates things like heart rate, the force of contraction of the heart, how hard it contracts, and vessel diameter. So we call that the cardiovascular center. It also contains the respiratory centers. That regulates breathing rate, control of respiration rate, the depth of breathing, how deeply you inhale and exhale. And it also includes the medulla, the vomiting center. When we have hiccups, is regulated or controlled by that part of the brainstem. Coughing, sneezing. So as you can see, all of these things, midbrain, pons doesn't have a lot of regulatory functions, more transmission functions, and predominantly the medulla regulates a lot of things that are sort of basic physiologic survival things like breathing, heart rate, digestion. I should have written that here, is also largely regulated in this area. Vomiting, we don't have conscious control over that. Hiccups, coughing, sneezing, all those things are just basic physiologic functions that we don't have conscious control over. All right, guys, let me cut my recording here and then I'll say a few more things.